Hello and welcome to this Teaching Ideas video on how to use a large model to help explain phagocytosis to either GCSE biology students or A-level biology students. Right, some of the key things to remember before you get started is to make sure that you have everything made well in advance. Right, split it up into as many stages as possible. As the shorter the stages, the easier it is for students to remember. When running through the activity, go through a stage at a time. Make sure that you're asking lots and lots of questions to students as you go through. If you've got parts to move around, for instance, the lysosomes within the phagocytes or the pathogens, you can get students to move them around. It's also another good opportunity to help reinforce the idea of antigens and the fact that specific binding sites will attach to specific antigens, just to help reinforce that with students, as the knowing about antigens is something that will often come up later along and is something that usually gets them marked. Right, the next short video clip goes through how to practically run through this activity during a lesson. In this short revision video, I'm going to be describing the process of phagocytosis. So, phagocytosis starts with a phagocyte being attracted to a pathogen. The pathogen produces small chemicals which attract the phagocytes to it. The second stage is that the pathogen is taken into the phagocyte by endocytosis. The phagocyte recognises the pathogen because it has non-human antigens on its cell surface. The third stage is that the pathogen is encased in a vesicle called a phagosome. Right? So this phagosome is a membrane-bound sac that encases the pathogen. The next stage is that two lysosomes within the phagocyte go and connect to our phagosome to form a phagolysosome. Hydrolytic enzymes and digestive enzymes and hydrogen peroxide is also now injected into the phagosome, which breaks up the pathogen. The broken up pathogen, most of it, is ejected by exocytosis. However, the antigens on its surface are connected to MHC, or major histocompatibility complexes, which move to the cell surface of the phagocyte. The phagocyte now becomes an antigen presenting cell. Hopefully this video has given you some ideas how to teach this particular concept. If you've liked this video, please subscribe, like the video. There's links below to follow me on Facebook, Twitter, and there's a link below to my shop on test where you can go and get a full copy of this lesson. Please feel free to leave any comments or questions below and I will get back to you as quickly as possible. Thanks for watching and happy teaching.